Good afternoon, everyone. Today I would like to present you my thesis project. First, I would like to start with a brief introduction about ocean acidification, also called the other CO2 problem. As you can see in the figure, when atmospheric CO2 concentrations increase, a decrease in ocean pH occurs. This is due to a higher CO2 uptake by the ocean, which changes the seawater carbonate chemistry. The current ocean pH is 8.1, but a decline happened since the beginning of industrial era, and a further decrease of 0.2 and 0.3 units has been predicted for the current century. These changes will impact marine life, and soft-bottom communities are not the exception. There are large animals that inhabit the sediments, such as many bivalves, but there are also other organisms much smaller, such as copepods and nematodes, that also live within the substrate. Now, I would like to focus on sediment-dwelling bivalves. In soft sediment habitats, bivalves can act as ecosystem engineers due to their feeding habits, mostly by deposit feeding, as they continuously rework the sediment surface with the inhalant siphon. Suspension feeding can take place as well, when they extend the siphon up to the sediment surface or to the water column. These activities create changes in pore water, which exert cascading effects in their communities related to disturbance and food availability. Bivalves mainly create in the surface disturbance, while in the sediment subsurface, they release nutrients, they create micro-oxyzones, and they increase microbial activity. Now, I have a question for you. Does ocean acidification affect bivalves communities? The answer is yes. Studies have found that it can affect negatively early stages of bivalves with a further decrease in their populations, but also positive effects have been found, such as the stimulation of food resources. The Abra alba community has been categorized as the most diverse macrobenthic coastal community in the Belgian part of the North Sea. This bivalve inhabits the fine sandy sublittoral, and it can be found in the upper 3 to 4 centimeters of the sediment. It has been classified as a biodiffuser, and it can exert suspension and deposit feeding. A previous study has found that they can exert cascading effects in their communities, such as the downward nematode migration and the depletion of food resources in the surface. Now, you must be wondering, does ocean acidification affect Abra alba communities? The answer is we don't know, which is why this thesis aimed to investigate whether the presence of Abra alba may alter ocean acidification effects on meiobenthos communities using control conditions. And I formulated this hypothesis to see if the pH, the presence of Abra, and their interactive effect had an effect on meiofauna density and its vertical distribution. Now the experimental setup. After subtidal sediment and Abra alba individuals were collected from a coastal station in the Belgian part of the North Sea, the sediment was sieved and mixed, and it was used to fill up 12 sediment cores. The incubation system consisted in two types of tanks, the header tank and the experimental tank, which contained two sediment cores, one with Abra alba and one without bivalves. Then the sediment cores were fully submerged in seawater with a natural pH of 8.1, and they were left to acclimatize to laboratory conditions for 4 days. Afterwards, the pH manipulation started, where 3 tanks were randomly allocated to the reduced pH, in which the pH was reduced 0.1 units per day during 3 days, by bubbling the seawater with CO2. And the other 3 tanks were bubbled with ambient air. Afterwards, all the cores were left to acclimatize to these manipulated conditions for 4 days. And then the experiment started. It lasted 6 weeks. On week 5, oxygen penetration depth measurements were performed, and the temperature varied between 19.3 and 19.7 throughout the experiment. At the end of the experiment, each sediment core was sliced on 1 centimeter intervals, down to 4 centimeters depth. Then each slice was homogenized and 2 subsamples were taken, one for meiofauna analysis and one for chlorophyll A. Afterwards, the meiofauna was extracted by centrifugation. The organisms were identified and counted. While for the chlorophyll A, it was extracted from freeze-dried sediment, and their concentrations were obtained by HPLC analysis. Regarding the data analysis, only data from the 3 upper sediment layers were considered, due to missing data in the deepest one. 2-way ANOVA tests were carried out to assess the impact of pH and the presence of Abra, while when determining differences between sediment layers, linear mixed models and generalized linear mixed models were performed. 2 key tests were used to assess differences between groups. And finally, a Mann-Whitney U test was used to see if the average surviving number of bivalves in the Abra present treatment differ between pH treatments. And regarding the survival of Abra alba, we did not find significant differences between pH treatments. The bivalves that were recovered from the subsamples, they were found between 0 and 2 centimeters, mostly located in the uppermost centimeter. Although copepods were very rare, we found significant effects for pH, regardless of the presence of Abra. No significant differences were found due to the presence of Abra or the interaction of both factors. Regarding the vertical distribution, we found significant effects for pH and depth, but not for the presence of Abra. Now, regarding the copepod densities, other studies have found a higher copepod densities under acidification scenarios. As you can see, in the reduced pH treatments, copepod densities were higher than in the ambient pH treatments. Previous studies have found that ocean acidification effects are species-specific, where some species of copepods may be more tolerant to cope with higher CO2 concentrations. Also, it has been suggested that copepods may be able to cope with acidification when excess food is available, and that they may reproduce as an initial stress response due to reduced pH. Now, I would like you to focus on the reduced pH treatments only. As you can see, lower copepod density was found when Abra was present. This could have been due to a competition between food resources between Abra and copepods in the uppermost layer, but it could have been also due to an interference competition related to deposit feeding. Regarding nematodes, among the meiofauna groups found, they were the most abundant ones, and not significant differences were found for pH, Abra, nor the interaction between both. Also, when analyzing the vertical distribution, no interactions between factors were found, and when assessing each variable, no significant effects were found for pH, Abra, and only a significant effect was found for depth. Even though these results, the profile suggests that the presence of Abra alba may be structuring nematode vertical distribution. Now, when Abra was absent, as you can see, higher nematode densities occur in the ambient pH treatment compared to the reduced one. Also, if we take a look at the reduced pH treatment, we can see higher nematode densities in this layer, in the uppermost one, suggesting that acidification could lead to a shift in vertical distribution. Also, as we can see, in the deepest layers, lower nematode densities were found. This could have been due to mortality. But if this were the case, we would see even marked decrease in both layers. And as this did not happen, this could have been due to a migration upwards to escape from acidified conditions, from this deepest layer to the upper ones. And unfortunately, we did not have a pH measurements for the sediment, although when comparing with previous studies with similar sediments, we can assume that the pH was low, below the 5 millimeters, due to DIC production in the upper layers. If this migration would have occurred, we would expect finding even higher densities in this layer, but this did not happen. So maybe species-specific mortality to low pH occurred in this layer. But but without a nematode community analysis, this is difficult to interpret because there may be species more tolerant to acidification. Now, what happened when Abra was present? Now, I would like you to compare these panels, A and C. And it seems that Abra alba had a negative effect on nematodes, decreasing their densities. While when comparing panels B and D, it seems that Abra favored nematodes under reduced pH conditions. How can we explain this? If we take a look to the chlorophyll A concentration profiles, we can see that when Abra was present, chlorophyll A concentrations decreased, but this decrease was prominent under reduced conditions. Also, and this could suggest a higher deposit feeding. Also, if we take a look at the oxygen penetration depth plot, and if we focus in the values when Abra was present, we can see that under reduced pH conditions, this value was shallower, suggesting a higher deposit feeding activity compared to the ambient pH, where suspension feeding may have occurred as well, generating stronger pressurizations in the sediment and increasing oxygenation. So this change in the feeding behavior of Abra alba may explain why we found higher nematode densities in the deepest layers. This higher deposit feeding would have increased the food availability by the excretion and also would have created less disturbance for nematodes by generating weaker pressurizations in the sediment. To conclude, reduced pH seems to affect copepods, but it does not seem to affect nematode densities. Also, even though we did not found significant effects for Abra, the profile suggests that the vertical distribution of meiofauna may be structured by the presence of this bivalve. This thesis also suggests that acidified conditions may change the feeding behavior of Abra alba, exerting cascading effects on the meiofauna and altering Abra alba communities. For future studies, it could be considered to perform nematode community analysis, as this cascading effect depends on the species and also on the feeding ecology. As I already mentioned, there may be also species more resilient to ocean acidification. And finally, it would be interesting to investigate the interactive effect of warming and ocean acidification to have a complete picture of the scenarios that could happen to these communities under climate change. I would like to thank my supervisor, Carl Van Collen, for this opportunity and for the guidance, and also to everyone else that was involved in this project. And thank you for your attention.